Thank you. And we're going to move to somebody who's, as I mentioned, really a pioneer in fly cancer development, one of the most innovative and rigorous thinkers I personally know, pushing this field forward. And so, Tano, if you can take over, it's all yours.
Okay. Let me. So I'm sharing already, correct?
Yeah, yes, you're good.
Yeah. Okay, so here we are. Fine. Okay. So, well, thank you, Ross, for those very kind comments. Thank you to the organizers, to you for the invitation to be here. Isabel Palacios has been asking me to join, which I would have been delighted to do several years back, but the timing never quite worked out well. And I'm delighted to be here, although sorry that it has to be under this limited format. We'll do our best so far. It's been fantastic. So these are the people in my lab, as you can see here, and this is the way we work on these days. And I also mentioned there the name of some of the people who contributed, not any longer in the lab, but contributors to the data that I will present today. Let me put things into context and tell you the reason why we went into these studies. Because at the time, we were actually not interested in cancer, not interested in asymmetric cell division of neural stem cells. Neural stem cells in Drosophila and in other species divide asymmetrically. This is one of those neural stem cell divisions, one of those neural stem cells, which is dividing, the chromosome sitting in the middle, and the division is strongly asymmetric. Every time the cell divides, generates another cell like itself, and one of those small cells that stops being a stem cell and enters a differentiation program. You can see all that happening live in videos we took years ago, a very highly asymmetric cell division of a neural stem cell producing two cells, one a renewed version of the original stem and another one, this one over here, which will differentiate. We can look at that from a more molecular perspective. And what you have here is the same cell, but now we have decorated the apical and the basal borders of the cell with certain protein complexes we know of. This is on the apical side, this is on the basal side. And this is just to let you know that the whole purpose of this highly sophisticated choreography of molecules being arranged in different parts of the cell is to ensure that all these guys sitting down here end up in the small cell and away from the large cell. This is the way it goes in real life. All those cell-fed determinants are transmitted to the cell that will enter differentiation, which in fact enters differentiation because it inherits those determinants. And when we were working on these issues, we came up with this question. Under normal conditions, when the red and the green complexes organize well, you get a normal brain developing. What happens when those processes, the processes of transmitting cell-fate determination during asymmetric cell division, do not work well? What happens to cells when neural stem cells and their daughters, when their instructions to continue the process of development are not perfect? And for that, we created flies, theristal larval brain, in which components of the red and green pathways were lacking or mutant. And we found that often the end result is this, a gigantic overgrowth of the affected brain. This is definitely a tumor, but not necessarily a malignant tumor. Finding malignant in flies is not such a trivial matter. So we came up with, we use the definitions that were put forward by workers before us, Elizabeth Gatev mainly, and we decided to assay whether this tissue, which is obviously much bigger and tumoral, has acquired the key characteristic of malignancy, which is immortality. And for that, we resorted to an ancient method of checking for immortality in flies, which consists in injecting this tissue under investigation into the abdomen of a healthy fly. I'm gonna show to you this highly sophisticated 21st century approach, which require incredibly high and developed technology, as you shall see in a moment. It takes a very sharp needle to pick up the tissue. Once the tissue is inside the needle, like you can see there, you come to some flies, put them on double-sided sticky tape, and inject. And that's the whole assay. There it is. The piece of tissue is now within the fly. And the question is, what happens now? Well, it happens that if you inject a little bit of this wild-type tissue over here, you can see the little bit right there. After 2 weeks, it's still sitting around and doing nothing because its proliferation potential is totally limited. However, if you inject the same amount of tissue doing the same procedure into a healthy fly, 2 weeks after the injection, the result is this. There is a dramatic, gigantic overgrowth of several hundred-fold overgrowth of the injected tissue that we start to characterize as malignant growth. And it's definitely malignant if we can then take a little bit of this tissue, as small as before, and reimplant again and again and again over the years. This proves immortality, and this is what we call malignant growth in flies. So once we stumble upon this kind of tumors, which is the way we found them, we decided to ask the obvious question of whether they also show some of the most characteristic features of tumors in humans. So one of them is the possibility that tissue tumor cells detach from the main tumor mass and are able to form colonies at distant destinations. And the answer is yes, indeed. You can see these colonies that originated in the primary tumor and are now someplace in the thorax and within the head. Within the fly, we look at it with more resolution. We can find this microfly type of metastasis in flies. It's a complex concept, but this kind of behavior we can definitely find. For those of you who don't work on flies, let me make clear that for these cells to reach this destination, they need to cross this peritoneal sheath that envelopes the ovary. This is the ovary of the fly, by the way, and then cross the muscle sheath, the epithelial layers, until it reaches inside the ovary. And so this is what we call invasiveness in this simple model. Another very interesting, very typical characteristic of cancer in humans is the presence of two types of instability, centrosomal and genomic instability. Centrosomes are these little two dots that stand here at each side of the mitotic plate of the chromosome metaphase plate. There are two per cell in humans and in flies. They organize the mitotic spindle, the microtubules. And it's been reported for over 100 years now that in cancer cells, the number and the structure of centrosomes are often abnormal. You see tiny little bits of centrosomes here and there, and at any rate, too many of them anyway. And those organize tripolar mitosis, which create problems we'll refer to later on. Drosophila normal cells indeed carry only two centrosomes, like all animals on Earth. And the question was, what happens to centrosomes in these tumors, which, let me stress, were not created by altering the centrosome cycle in any way, by altering asymmetric cell division of the neural stem cells. And what we found is that, there are centrosome instabilities as well. A significant fraction of the cells present abnormal centrosomes numbers and also centrosomes with abnormal structures. The next thing we checked was for chromosome instability. You are all familiar with very abnormal karyotypes found in cancer cells. This is a very common feature of most human carcinomas. This is the wild-type karyotype of flies, only 4 pairs. The wild-type karyotype of humans is a few more pairs, but of course it's very rigorously maintained from cell division, during cell division, and all cells present the same characteristics. Human cancer cells display very abnormal karyotypes, and we found that so do most of the brain tumors that we can generate through different methods. And so this led us some years ago to define what we call the most common distinct traits of Drosophila malignant brain tumors, which are that cells become immortal and, very importantly, acquires limitless replicative potential, and be also extremely important, these cells become irresponsive to differentiating signals. We haven't shown you that, but we can do tests for that, and these cells do not respond. They will never go through the processes that normal cells will go through during puberty formation, for instance. They will just keep dividing aggressively and malignantly. These cells can also detach from the main tumor mass and colonize some tissues, what we call fly metastasis. And very interestingly, a fraction of these cells present aberrant centrosomes and aberrant chromosome numbers, chromosome instability, which are features found often in human carcinomas. Now, having those models, we decided to start asking questions, use the model to address very simple questions regarding human cancer. And one of the very first one we decided to address is here. It refers to Bovery's hypothesis of tumorigenesis, which was put forward 100 years ago, more than 100 years ago, and is still under debate in humans. We address this issue in flies as follows. Bovery's hypothesis claims that the origin of tumors is the presence of too many centrosomes, 1, 2, 3 centrosomes here. And too many centrosomes create multipolar spindles that results in aneuploid cells that are transformed and overgrowth. And the reason for that is what I showed you before, that cancer, that tumors, human tumors, very often, cells within those tumors, very often are tripolar, tetrapolar, present abnormal contents of centrosomes and chromosomes. So his claim was extra centrosomes cause aneuploidy, which cause cancer. Like I said, still under debate. There is strong evidence in favor and against in human cancer. We address these questions in flies by the very simple approach of creating larval brains that carried more than 2 centrosomes, and also larval brains that carried larval brains which contain cells that carry more than 2 centrosomes, and also others that contain cells with no centrosomes at all. And what we found is that, as opposed to control implants, which none of them resulted in tumor growth, when brains carry cells that contain more than 2 centrosomes, 164 injections here, about 27 of them, many cause tumors. And interestingly, even without any centrosomes, tumors were also present. This actually was the first demonstration that abnormal centrosome numbers are tumorigenic in flies. All right, but go back to the original call. The original claim is that extra centrosomes cause cancer because they create aneuploidy. And so if that is true, centrosomes, abnormal centrosomes, too many or too few, should be tumorigenic in any tissue in the fly. And so we did the same assay in 30-star imaginal discs. And the results are here. Out of several hundred transplantation experiments, none of the cells containing more than 2 centrosomes, none of the tissues with cells containing more than 2 or no centrosomes produce tumors, which strongly suggested that extra centrosome numbers or loss of centrosomes altogether is a tumorigenic event, but a rather cell type specific tumorigenic event, which doesn't quite go well with this call summarized here. To study that further, what we did was to create cells conditions in which cells carried abnormal chromosomal numbers, aneuploidies of different types, polyploidies, small aneuploidies, and so forth. And just to summarize that, all that means is injections of wild-type brains, which carry the wild-type karyotype that you can see here, and injections, allograft assays with tumors, with brains that were mutants for different components, and therefore carried abnormal karyotypes of, like these ones you can see here, of different kinds. And the result is here. By altering spindle assembly checkpoint and other components of chromosome segregation, we create aneuploidies. By altering cytogenesis, by mutagenizing with X-rays, we create different kinds of chromosomal abnormalities. We perform all these injections, and the result is very simple to interpret. Chromosome instability is not creating tumors in this model system, and that led us to revise Bovery's hypothesis, because on the one hand, we have proved that abnormal centrosome numbers, like he claimed, result in overgrowth. But what we have observed is more compatible with interpretation that these abnormal centrosome numbers are tumorogenic in tissues where some cells divide asymmetrically, like the neural stem cells, and then the lack of centrosomes or too many centrosomes alter asymmetry in different ways. This red component is lacking or not well localized, whatever, call it a name. And the lack of proper asymmetric cell division is what results in failed cell fate determination, which eventually results in tumor growth. And so that has taught us a few lessons from the models we work with. One of them is that centrosome dysfunction is tumorogenic indeed in neural stem cells, but not in imaginal T cells. And that aneuploidy chromosome instability, CIN, is not tumorigenic in neural stem cell or in imaginal T cells. And from there, we can conclude that the tumorigenic effect of abnormal centrosomes is definitely cell type specific and is not due to the resulting genome instability and could be due to the resulting disturbance in cell fate. We also wonder about genome instability, lesions in the, I read sessions, it should be lesions in the genome, damage we introduce in the genome in terms of single nucleotide polymorphism, deficiencies, translocations, and so on. And what you have here is what we know from human cancer, from rhabdoid tumor to melanoma, the number of single nucleotide polymorphism per megabase. One take-home message, please note that this scale is logarithmic. You may believe, like we all do when we start in this field, that genomic instability and cancer come together, but it is not necessarily the case. It depends on the type of tumor. Some tumors present very low levels of genome instability, others present very high levels of tumor instability. This is true. But even within those, like lung adenocarcinoma, which in average presents a very high rate of mutation, individual cases range from 100 to less than 0.1 single nucleotide polymorphisms per megabase. So the distribution is very wide. And so what we did was to full genome sequence our tumors and find out to what extent Drosophila tumors look alike. And the result is, yes, they do. They spread well within the range of the level of single nucleotide polymorphism observed in human tumors of many different kinds. This is for single nucleotide polymorphism. We have done the same for other type of lesions, like duplications and deletions, and to cut it short, the same applies. The range, the size, the distribution of different parameters of deficiencies and duplications, generically referred to as copy number variations found in Drosophila tumors, are well within the range of what we find in human tumors. However, let me show you this in this conclusion. The frequency of mutations as well of those as the frequency of deficiencies and duplications, copy number variations is the way they are normally called. In Drosophila tumors is well within the range of observing human tumors. However, I was saying, lesson number 5, none of those out of the hundreds of copy number variations that we detected in Drosophila tumors and hundreds of single nucleotide polymorphisms that we detected in Drosophila tumors, none of them correlated tightly with tumor growth. That is, none of them behaves as driver. You can find all sorts of combinations and some drivers which are accumulated at certain stages of tumor growth then disappear with no effect whatsoever on the tumor, which continues growing, and so on and so forth. So there is a very significant level of genome instability in general, but no driver that meets the definition of a tumor driver was found within this type of tumors that we are investigating. Okay, so let me change gear to a different tumor type we have been working on over the years and has also taught us some lessons about tumor growth. We call it for short MBT. These are also tumors that develop in the brain. And a very relevant piece of information we obtained from these tumors came from plotting the level of expression of the transcriptome of different individuals. This is MBT tumor number 1, number 2, number 3. And what you have here is the heat map showing those genes which are downregulated with respect to wild-type sample in blue or upregulated with respect with wild-type samples in blue. There is a lot of information to derive from there. But for the purposes of today's presentation, I just want to draw your attention to this group over here, which includes germline genes. These are genes required for oogenesis or spermatogenesis. Without them, oogenesis or spermatogenesis doesn't work well, do not work well, but not normally expressed, not expected in somatic tissues of any kind, definitely not in normal brains yet. They are very highly upregulated in MBT tumors. Let me tell you, because that's the way it is, that when we found this, the first thought was that flies are very weird, that the kind of tumors that you may generate in flies are peculiar, and so on. But then we also add that all that way of thinking came out of sheer ignorance. Because when we went into the literature, we became familiar with literature that had been published over the years. And this 2005 review is a good summary of work carried out over the previous 2 or 3 decades, referring to something that was unknown to us at the time, called cancer testis antigens. And I just summarized, presented for you 2 other papers, later, more recent papers. There are hundreds of these, because this is a very wide and important field in oncology, the field of cancer testis. What are cancer testis in human oncology? As defined by human oncologists, genes that are normally expressed only in the germline and become ectopically expressed in somatic tumors of many different types. That is quite reminiscent of what we found. And for years, and still under discussion, people on oncology, human oncology, real oncology, have been discussing whether or not there are reasons to be in favor, there are reasons to dispute that they could contribute to malignant traits like immortality, invasiveness, immune evasion, and so forth. In humans, these genes are given a tag name. It's called the CT number, cancer testis. And let me make clear here from here onwards that cancer testis is the classical name for them. A better name, a more appropriate name is cancer germline, because some of these genes are specific of the ovary actually. So cancer testis, cancer germline genes, synonyms for the purpose of this presentation. Like I said, all of these human genes are given a cancer testis number. And so we went back to our data, and to our delight, we found that out of those genes, and these are just a few of the ones we found, some of them were actually the homologue, in some cases, the functional orthologue of genes that in humans have been decades before we made this observation renamed as cancer testis number something, like cancer testis number 8 for SYCP1, which is the human orthologue of C3G. So with this tool in hand, we decided to, as far as flies are concerned, address the question of whether or not germline genes are actually, these germline genes which are regulated in cancer, are actually required for tumor growth. And this is something we did some years ago using the very simple approach shown there. A wild-type brain normally developed, which upon injection cannot overgrow. An overgrowth, largely vastly overgrowth brain tumor caused by the mutation in MBT. Upon injection, it grows and kills the host. very fast. And now we wonder, what if we make the flies mutant for MBT, the tumor should grow, but also mutant for PXT. PXT is the most highly upregulated protein in MBT tumors. It's upregulated several orders of magnitude. It is a dramatic case of upregulation, I can tell you that. And so what if we remove that gene? What happens to tumor growth? And the answer is nothing happens. This is the most highly upregulated gene in the tumor, but the tumor could not care less. It grows just as big and it is just as malignant. And so what about others? This is a long story because we're talking dozens of genes. Let me show you one interesting example. We make the flies mutant for MBT, tumors should grow, but we remove these germline genes, nanos, and then the brain cannot grow. And the brain cannot grow, and if we inject the brain, it just cannot grow. So nanos, which is a germline gene, is behaving here as a totally, absolutely required component of MBT tumorigenesis. In that sense, it's a suppressor of tumor growth, removing nanos impedes tumor growth, which allow us to draw this very simple cartoon, this very simple scheme. We have known, actually we didn't, but we learned through the literature that human oncologists knew that human cancer often happens, correlates with the expression of germline genes, and we're talking somatic cancer, cancer in somatic tissues, often overexpress germline genes, and that correlates with malignant growth. And this discussion was going on when we entered this field, and it's still going on to a large extent in the human oncology field. But using our far more simple approach, we were able to address this question. And first of all, we found a tumor in flies in which the same thing is true. It upregulates germline genes. And in this particular model, we were able to set the arrow in the right direction. And yes, indeed, some of these genes are absolutely essential for malignant growth. I will derive a third list of lessons from there, which is some Drosophila brain tumors can upregulate cancer germline or cancer testis genes, whose orthologs are actually cancer germline, cancer testis genes in humans. The importance of this observation is that Drosophila can be used as a model to study the role of those genes in malignant growth. And what we have found so far is that some of those conserved cancer testis genes are dispensable, yes, but others are absolutely essential for tumor growth. And this is really important because probably I'm sure you all have already come across this idea and have reached this conclusion. These are ideal targets. These are genes whose side effects, the worst side effect would be sterility. Killing these genes in flies, for certain, impedes tumor growth, causing as a collateral effect sterility. No, it doesn't affect longevity. It does not affect viability. It's a totally bearable side effect for most malignant human tumors. So with this information in hand, we decided to use this model to interrogate the genome, the entire genome, not the entire genome so far, but 4,000 genes, which is a very good chunk of the entire genome, to carry out an unbiased screen to identify genes without which the tumor cannot grow. And we want this to be unbiased. We could have, and it's very important, and we are very much in favor of that kind of approach, we could have do a mini screen looking for the importance of Notch and P53 and you name it. That is something we are also doing actually, and it's very informative. But here we are using flies for what they are really, really powerful for, which is to run into the unknown, to discover what you wouldn't even think of, and that has to come through unbiased approaches. So without knowing anything about biology, let's just inactivate one by one each one of these genes and see whether which of them is essential for tumor growth. That means we have a tumor, a BT tumor, highly neoplastic, much larger than the normal brain. It will grow much bigger than this later on, and highly disorder in anatomy, compared to a nice, normal-looking Walton brain. And again, to cut it a very, 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 to cut a very, very, very long story extremely short, I will just show you one example. From there, we got two types of suppressors. One, let's call them suppressors type A, impede tumor growth. The brain actually doesn't grow big, actually. It reaches this size, which is even smaller than the wild type, and it doesn't go any further. This is good. Without these genes, there is no tumor growth. We can discuss about how applicable that could be, but let's leave it there. They are potentially interesting genes. But of course, even more interesting than those genes are what we call suppressor type B, which look like this. What you're looking at there is a fly, which is a brain, which is MBT, and therefore the MBT tumor looking like this one is scheduled to grow, but the fact that it's lacking one of those suppressors B type means that the tumor, not only the tumor has not developed, but instead a totally normal looking wild type brain has. And one of those is known by May W68. This is what unbiased screenings can give you, because there is no way I would have thought of trying and see whether this gene affects tumor growth, because this is a gene which is specific of the female germline, and it's only required during meiosis. And what it does is to induce the double strand breaks that you need to introduce in homologous chromosomes for recombination to take place. This is the enzyme that makes double strand breaks during meiotic recombination. If you kill this enzyme, the flies are perfectly viable. They're perfect, but females cannot, there is no meiotic recombination. In the case of Drosophila, this is female specific. And yet, when this gene is lacking, the brain, somatic tissue, cannot develop MBT-looking tumors. This is MBT, this is the wild type, this is another staining for another marker. This is relevant, a way of highlighting another structure within the brain, which is highly distorted in MBT. And this is a very important marker. This is VASA. This is pure germline. Absolutely not to be seen, with a few exceptions we can discuss there, but definitely very low and almost absent in the wild type and highly upregulated in MBT tumors. This is the way MBT looks like, and this is the way MBT looks when you kill this critical gene for MBT growth. And actually, you could actually raise the issue of whether this is a double mutant MBT May W68, as I am telling you, or a contaminant, a wild type fly that happened to invade our culture. And what we're looking at here is a wild type fly. And no, it isn't a wild type fly, because you notice still expressing the germline gene. So this MBT is upregulating germline genes, many of them, and certainly VASA. And yet, because it's lacking only this gene, it cannot grow, which actually opens the very interesting question of what happens to other germline genes when you remove the tumorigenic potential of MBT, when you make double mutants like MBT May W68. This looks anatomically like a wild type. Upon injection, it cannot grow. What happens to the yellow genes I showed you at the beginning, the germline genes and others, those genes that you can see here, which are highly upregulated compared to the wild type? I'll give you 1 second to bet on it, and after 1 second, I'll tell you that most of them are still there. This also carries a very interesting message, which I think I have summarized, and I will tell you at the end. Let me just add to that these are some of the hits, some of the genes that we identified as tumor suppressors. You cannot go through that, it's too small. This is published, you can find out about these genes. Let me just highlight 2 of them. One of them is TCTP. I highlight this one because TCTP, which stands for translationally controlled tumor protein, is one of those genes that is highly conserved to the extent that the expression of the human gene in flies rescues the fly phenotypes. That's an interesting gene. And on top of that, it's extremely interesting because it's one of those proteins which is known to disappear not one of those, but among the very first, if not the first protein, which starts to be downregulated in the case of human tumors when they regress. Some childhood cancer types present certain levels of spontaneous regression. The tumor grows, the patient's survival is highly compromised, but eventually, for unknown reason, the tumor disappears. TCTPs are very good markers for those tumors, and one of the very first molecular signs of tumor disappearance in human subjects is the loss of TCTP. Turns out that the loss of TCTP is a dramatic inhibitor, a very potent inhibitor of tumor growth in W68 tumors, which results in wild-type looking MBT, in MBT tumors, I meant to say. MBT tumors do not develop as MBT tumors upon loss of the TCTP. And not only that, when the tumor is MBT, when an MBT tumor is lacking the TCTP, it looks like the sample I showed you before, very much like a wild type. Another case is the case I already mentioned, MAW68. I'm going to give you a little additional piece of information, which is MAW68 has a highly conserved homologue in humans, which goes by the name of SPO11. And way before we made these observations, SPO11 in human cancer was referred as cancer testis number 35. So way before we found that through our unbiased screening, that the loss of this gene impedes MBT tumor growth. It was known in human oncology that the ortholog of MAW68, human ortholog, SPO11, the recombinase require the DNA the enzyme that breaks the DNA to allow for recombination during meiosis in humans is also expressed in a large number of human somatic cancer types. And so a few more lessons from there. The eighth is that unbiased genetic screens in Drosophila have identified genes which are known to be tightly linked to human cancers, and that provides some type of validation for this kind of studies. And of course, what is even more interesting, all this that were totally unsuspected. And going back to the data I showed you before, another conclusion that we can derive from this observation is that unscheduled upregulation of gene expression, upregulation in general, the fact that a gene is highly upregulated in a tumor sample versus a wild-type sample means close to zero as far as functional requirement is concerned. We have numbers for that. So we know, for instance, that 15%, or let me put it this other way, 85% of the 100 most upregulated genes in MBT are absolutely dispensable for MBT growth. Okay, so let me just finish by giving you two more examples of lessons derived from MBT.
You can wrap it up.
Yep. One of them is the fact that it is highly dimorphic, sexually dimorph. We came across this through studying MBT through the confusion derived from looking at MBT phenotypes. You can see there are many examples of different MBT tumors, and as you can see, they are highly dimorphic. To finish this observation, eventually we were able, through imaging this, looking at these images from a different perspective, find out that this variety of phenotypes can be assigned to two different sexes, depending on different components, the male and the female. Let me stop here for a second, because something came. I think it's Ross telling me that my time is up. Ross?
Yes. Yeah,
okay, fine. Sorry, I have a timer here. I'm completely confused. Sure, no problem. Fine, no problem at all. I understand. I don't want to take more time than allowed. So, well, fine to leave here. Let me just conclude from here that we use this dimorphic phenotype in MBT to explore something that we call the sex-specific tumor signature, and that just take you to the concluding slide from here, which is through studying this MBT tumor, we were able to identify something which is rather different to the standard signatures. It's a very simple view of healthy tissue in green transforming to malignant tissue in red. And that actually, as you know, correlates with the presence of tumor signatures you are all familiar with, that is genes which are up or down regulated in tumors compared to the healthy tissue. And what through our studies we are finding is a different type of tumor signatures in certain tumor types. And this is also very important in human cancers. In our tumor types, what we are finding are sex-linked tumor dimorphism signatures, which refers not to difference not between tumor and normal tissue, but to tumors tumors which develop in female samples, in female individuals, and tumors that develop in male individuals. There are some genes which are highly differentially dysregulated, and in some cases, some of these genes are responsible for the different degree in malignancy. In this particular case of the fly tumor we work with, males are more aggressive than females, and some of the genes which are responsible for this exceeded additional level of aggressivity, of malignancy, belong to the signature, the group of genes that are highly upregulated in males compared to female tumor samples. And I will leave it just there. Thank you very much for your attention.
Excellent. Many thanks, Tano. And as always, we have a burst of questions. And so to keep us on time, I'm actually going to ask sort of a larger picture summary question, and then I'm going to put the rest of them on Slack, okay? Because there's a lot. And in fact, Miriam Adeniki Saladin says that she's not into cancer research, but man, my notepad is filled with a lot of interesting info. Nice work, Professor Tano. I think that kind of summarizes it. That was amazing. So let me just ask you a big picture question, which sort of draws from some of these, and that is thinking. You probably, I know because you and I have talked a little bit about this. You probably thought as much about the future of fly cancer research as anybody. So where do you see the central contributions over the next 10 years that DrosAfrica can contribute to cancer research?
Okay, well, I would say this is the kind of question you ask in a job interview when you don't like the candidate and you want to make his life miserable, right? But yeah, I'm sure this is the question. I completely agree. Well, I don't know, but I'm happy to tell you what we would like to do. We would like to keep exploring the unknown, the try and continue it's an excellent question. Let me start again. There are many ways. I only given you people Ross knows only too well because he's part of this. We're doing this approach, which I won't repeat. I told you already. Other people are using totally different approaches, and all of them are more than welcome. All of them are working very well. Ross is an excellent representation of a completely different approach. Use the fly to recapitulate literally what happens in humans, and use the flies as little assay test tubes to identify therapeuticals and other things. This is one way, creating flies a la carte, putting there the mutations which are known to operate in human tumors and use flies to do what you cannot do in human tumors. Fantastic way of going for it, working exceedingly well check the papers. It's already published, most of it, and not most, but a good chunk of it, and a lot more coming for sure. And then there is the other way, and probably many other in between, many others in between, which is to use the flies to answer, to approach very naive questions. So the Bovary hypothesis is boring. I mean, if you go into the literature, just go for it. Go to Google, write down Bovary hypothesis of tumorigenesis. Oh yes, I mean, 50% yes, 50% no. We don't know. I cannot tell you. Let me just say, every time I give a talk, I always say this 20 times. We don't know anything about human cancer, nothing at all. But what I can tell you is what we are discovering in flies. And as far as flies are concerned, too many centrosomes are tumorigenic only in the brain, not in any other tissues which divide methodically. And I don't want to repeat my seminar here. And so we want to continue this kind of approach, and I think there is plenty of room for maneuver. And very particularly, to be a bit more specific, I think there is great potential still unexplored in the cancer testis field. I was really delighted. Let me tell you this. There is a paper out there in which what they did was to use the germline genes that we found upregulated in Drosophila tumors. These are human oncologists, and they look for the human homologs of those genes and look for the expressions of those genes in human cancer. And through that reverse approach, they were able to identify another 20 new cancer testis genes that were totally unsuspected before, only because they are the homologs of the ones we found that were completely unexpected. So as far as I am concerned, I very much would like to continue exploring, and there are many ways of doing that, the group of cancer testis genes. And I don't have time to go into that. I would just leave it there in case any of you is interested. Happy to discuss it. I don't have time to make another 1-hour presentation claiming with very good pieces of evidence from my lab and from others that these Drosophila tumors recapitulate particularly well childhood cancer in many different regards, quite tantalizingly so, I would say. And so we're actually going into that. We're already collaborating very closely with oncology group, human oncology pediatricians nearby, and I think there is also good prospects on that line. Sorry, too long an answer, but too difficult a question.
But that will work. Hey, listen, Tano, much appreciated as always. And everybody, we're going to take a 15-minute break. We'll be back in 15 minutes, so 5 minutes after the hour, to hear Laura Towert, who has a fly company in the UK. This should be really fascinating. So everybody get up, take a stretch, eat some food, come on back. Let's do some more fly science.